Hello there. We've got a rather packed lecture ahead of us. Enzyme kinetics, which is the study of how enzymes work and how fast they work, is quite a rigorous topic, but I think it's really interesting and it's really important for biochemistry. So I hope you enjoy it as much as I do. The learning goals for enzyme kinetics will be in tandem to derive the mathematical underpinnings for measuring enzyme activity, while we strive to understand the assumptions needed to simplify and apply those Gen Chem rate laws to enzymes. Our molecule of the moment will be acetamide. The question that I have paired with this may look intimidating, but once you put pen to paper, I predict it will start to look familiar. Make sure you work on this and come ready to present when we start lecture. I like to set the stage for enzyme kinetics by talking about the reaction coordinate. You can learn a lot about what enzymes do and what they don't do based off of this plot, this visualization of the energy changes that occur as we transform our reactants into products. Let's recall that the activation barrier of a reaction will be the energy difference between the reactant states over here and the transition state all the way up there. The y-axis of the reaction coordinate is free energy. So the change in free energy of this uncatalyzed process is going to be very, very large. This is a very steep hill for our reactant or substrate to climb without any help. So what the enzyme does, it's going to give us a shortcut, a tunnel through this mountain that will help us get to the other side. That tunnel, shown as that blue curve, corresponds to the reaction coordinate of the enzyme catalyzed process. Notice how the activation energy between the transition state, the highest energy points of the blue curve, between the reactant state of E plus S is much smaller than the uncatalyzed process. This is a nice visual on how enzymes lower activation barriers, which in turn catalyzes the reactions. It speeds them up and makes that hill easier to cross. In a more numerical sense, what we're doing is increasing the reaction rate constants of that process. You want to know what the presence of that enzyme E does not change? It does not affect the change in energy between the reactant state and the product state. Both the red uncatalyzed coordinate and the blue catalyzed coordinate end at the same energy level. The delta G of the reaction, which is just the energy difference from here to there, isn't going to be affected. It's a very big misconception that the deeper this delta G sinks, the faster the reaction will go, and that's not true. The speed of the reaction, the kinetics, is solely dictated by activation energy. Said another way, enzymes do not alter reaction delta G. Another important fact about enzymes is that they aren't changed or consumed permanently by the reaction that they catalyze. They will guide our substrate through that shortcut through the mountain, that mountain tunnel, but they are going to be completely unaffected by it. And they are happy to go back to the beginning and chaperone another substrate along that shortcut. And the way that enzymes persuade the substrate to go through this shortcut is by binding with them. Generally speaking, enzymes will bind a specific set of substrates and be selective in which compounds they will guide through this accelerated reaction path. You can see that right over here. Take a look at that, ES. That stands for an enzyme substrate complex. Here we have enzyme and substrate docked together, and what happens to the delta G? It goes down. Here, what we're seeing is that the enzyme substrate complex is stabilizing sort of the substrate. It's saying, here, come this way, we're going to make it easier for you, and that's going to get the ball rolling down this pathway. A second very big misconception about enzymes is that yes, even though they bind substrates, they are technically not great substrate binders. They're decent substrate binders. What they really are, they're good transition state binders. They're going to stabilize the transition state, and that's what brings this down so far. There is an energy dip over here, but it is marginal to the energy dip observed over there. Shown here is a very famous experiment that demonstrates how forming the transition state easier is what speeds up chemical reactions. If we consider a very simple anhydride synthesis between these two compounds coming together bimolecularly, into this anhydride, they will do so at a certain rate, which we will define as 1 for the time being. What happens if we did the same experiment, but we made it a unimolecular reaction? We can do that by anchoring these two carbons to each other. What have we done here? We restrict the ways that they can collide, and thus we increase the rate by a factor of 10,000. If you repeat the experiment and you restrict the degrees of freedom even more by attaching them to this bulky ring system, guess what? The reaction rate will increase even further. There's a word for this. It's catalysis by approximation. It's reducing entropy. This experiment, I think, does a great job of visualizing how reducing substrate entropy drives the formation of the transition state, easier at least. So enzymes drive reactions by, first and foremost, in my opinion, reducing substrate entropy. Besides reducing reaction entropy, I'll just write that again here. The next way that enzymes help substrates react is by binding them. Substrate binding achieves two things. One, enzyme binding sites typically run hydrophobic. This is important because that hydrophobic cleft will strip the solvation shell, protecting the substrate from other reactants. All right, that's got to go. Desolvating substrate. Secondly, enzymes don't just bind the substrates, they will actually distort them a little bit to help the substrate approximate the reactive transition state. And that's really a key takeaway here, the distorting of the substrate. Enzymes by design are not amazing substrate binders. And we can look at these illustrations to the right to help clarify that. We will imagine this fictitious enzyme called stickase. It's an enzyme whose job is to break imaginary sticks. The substrate, that'll stick, will react like so without a catalyst or enzyme present. We have our stick, we put in a lot of energy and squeeze it into a bent reactive transition state. And once we've stressed this part of the metal, it'll snap. And that will give us two products, the halves of the broken stick. Now, let's imagine we had an enzyme helping us, and the enzyme was a really, really good substrate binder. Its pocket was a perfect match for the reactant states. We have our enzyme, and let's line that pocket up with magnets and make it really attracted to that metal stick. It's going to wiggle in there, and look, it's really stable and really happy. When things are stable, energy goes down, and we're trapped in an energetic well. We're happy here. We don't want to leave here. And as a matter of fact, leaving here towards the product side of the coordinate has actually become more difficult. We made the mountain taller because we don't want to leave the magnets. So this is a good visual as to why enzymes aren't really meant to bind the substrate perfectly. This last illustration does a great job showing why enzymes are okay substrate binders. Great transition state binders. Here, we will sneak in our metal stick into an actually useful enzyme, shown in C, and look, we are making contacts with magnets, so the enzyme is able to reel in the substrate decently enough. But we're not done yet. The magnets all over here on that inner curve are coaxing that stick to bend and adopt this uncomfortable transition state. And that's how the enzyme helps the substrate go in the direction of the products. It will persuade it to bend and then snap. That's the last key takeaway here. It's all about stabilizing the road to the transition state. A quick point of order before we move on. Sometimes students confuse the following terms: intermediates and transition states. Transition states are energetic maxima. They are going to be the top of any kind of hill shown. These are not isolable. They form and break in an instant. They contain partially broken, partially forming bonds. They're not intermediates. Intermediates in a reaction coordinate are energetic minima. This dip over there is an energetic minima. It's an intermediate. You can isolate an intermediate. Those will have fully formed and stable bonds. They may be higher energy, perhaps, than the reactants, but they're actual chemicals. you can't directly observe a transition state. Just be aware of that. Enzyme kinetics is all about rate and mechanism. You may remember from general chemistry that we can bin reaction rates into one of three orders. Let's recap them briefly right now. Zero order means the reaction speed is unaffected by the reactant concentration. Adding more reactant does not make it go any faster. A first order reaction means the reaction speed goes up as the reactant's concentration increases. Second order means that the reaction speed will go up with the concentration of reactant squared. Now consider this plot. To a solution with a fixed enzyme concentration, increasing the substrate concentration in the x-axis at first will cause the reaction velocity or rate in the y direction to increase. The increase isn't exactly linear though. As we keep on adding in more and more substrates, that rate of the reaction begins to taper. It will actually approach this so-called speed limit asymptotically called the Vmax. Now ask yourself, what is the reaction order of an enzyme catalyzed process? At first blush, you may think it's first order because increasing the substrate increases the reaction speed. Sure, but later on in the plot, the reaction order looks more like zero order kinetics because now the speed isn't affected by that excess reactant, not dramatically at least. This is a really tricky question to answer. Enzyme kinetics don't fall neatly into any one category, which means there's no simple one equation that can tell us what the rate is for a given substrate concentration. Or is there? Our goal for the next few slides is to find a way to create this all-in-one reaction rate law for our enzymes. To do this, we need to make many simplifying assumptions to make this goal easier to achieve. Our first two assumptions are simple. First, assume one enzyme binds one substrate, and it will convert it into one product reversibly. 1E binds 1S to reversibly form 1P. Yes, I know some enzymes may combine two substrates together into one product, or it takes one substrate, breaks it into many products. Forget about them, all right? Let's also assume we will be measuring enzyme rates in a test tube under easy, stable conditions. We will assume that the concentration of enzyme is constant. There are no cells in that test tube making more enzyme or eating up enzyme and throwing off our calculations. We will also assume that we added in a vast excess of substrate relative to the concentration of enzyme, like 1 enzyme for every million substrate molecules. So that after we watch this reaction for like an hour, let's say, we will not have depleted our substrate by a significant percentage. We can combine these two assumptions into a single reaction equation. E plus S will reversibly form an enzyme substrate complex, and that bound enzyme substrate complex will simultaneously disassemble and react to yield free unchanged enzyme plus product. I'm putting in double arrows because we are again assuming reversibility. And each one of those arrows will have a rate constant because it will occur at a certain rate. So I will arbitrarily call this K1, which means the reverse process is K minus 1. This is K2. The reverse of K2 will be K minus 2. All right, already this is looking a little messy. Let's try to use some more assumptions. Our third assumption will be that we will start with no product in our test tube, which means we are only measuring the initial velocity of the reaction. If there is no product, that means K2 cannot happen. K minus 2, excuse me. Not after a very, very, very long time at least. So this is really convenient. We can simplify our expression like so. E plus S forms ES, forms irreversibly E plus P. We are just dropping K minus 2 from the expression. Let's make one more assumption. We will assume that the enzyme is either a free enzyme or one bound within the enzyme substrate complex. Those are the choices. E total equals free enzyme plus bound enzyme. You may be thinking, well, I guess that's sort of obvious, right? Why do we have to spell it out? It'll make sense in a little bit. Our third assumption was nice because what we essentially did was eliminate one of these 4 variables we have to solve for. But we still don't exactly have an equation yet, and that's where the 5th assumption comes in, which is perhaps the most important. We will be assuming that the enzyme substrate complex is a constant. And for that to be true, it means that the rate of formation must equal the rate of its breakdown. The word for this assumption is called the steady state approximation. And notice how it's in pink. If it's pink, it's important. And I even put it in all caps. This is a really big deal. And it implies a lot. Writing it out in terms of math, it means that the rate of formation equals K1 times the concentration of E times the concentration of S. And it means the rate of breakdown, which are the processes that are going away from ES, equals K minus 1 times ES plus K2 times ES. And since this and this formation and breakdown equal each other, we can glue these two equations together like so. We will get K1 times E times S equals K minus 1 ES plus K2 ES. The steady state approximation is really neat because it implies that K2 will exceed K1. The formation of ES will be the slow step, and the formation of products will be faster. It means like the moment we form enzyme substrate complex, it falls apart to product. It falls apart to product. That's how we're able to keep this ES steady. Said another way, it will never accumulate. Now, there are a few problems with this equation we got out of our steady state approximation. First is we don't know any of these rate constants. We still have three variables. Not good. Even worse, we don't know how much of our enzyme is free and how much of it is ES. So what we can do is rearrange our equation bit by bit until we have reaction rates on one side and a bunch of terms that we do know on the other. And the first fix to this problem is to get rid of E. Think back to our fourth assumption where we said total enzyme equals free enzyme plus ES. Well, you can rearrange that like so. And get the following expression. The leftover enzyme E equals the total enzyme minus the concentration of the enzyme substrate complex. Doing the following substitution eliminates the variable E, but it does make our math look uglier. The good news is, though, we know what E total is because we measured it in our theoretical experiments here. It's the amount of enzyme you dissolved in that test tube. To recap, our previous manipulation did eliminate that pesky E term. However, it came at a price of making this equation hideously complex. And I don't like math. So how do we make this even simpler? For starters, we can take these K1 and S terms and factor them into this parenthetical on the left side of the equation. And on the right side, let's do the opposite. There is a redundant ES term that we can factor out. So what does that look like? I'll just write over here that K1 times ET times S minus K1 again times ES times S equals K minus 1 plus K2 all multiplied by ES. And I know what you're thinking. Ew, how is that any better? Well, a theme with derivations is that sometimes you got to take a step back to make 2 steps forward. And this is how you do that. We will divide both sides by K1. And look what happens when we do that. I'll just draw a line over here, squeeze in a K1, and oh look, there's a K1 term now on both sides of this complicated binomial. So all of these go away. But we have to move it now into the denominator of this other term, like so. And since we're assuming a steady state and the rate constants of k minus 1, k1, k2 are not changing, what we can do is combine these 3 unknowns into a giant mega constant. And what will that look like? It will look something like this. We can call it, I don't know, Km. And over here, I have shown all of the unit cancellations that occur between the k minus 1, the k2, the k1 constant. It works out to have a unit of just simply molar. And that's important. Keep that in the back of your mind. And let's apply that to our rearrangement that we just did. It will simplify into ET times S minus ES times S equals Km times ES. You will perhaps have noticed a theme developing by now. We are trying to get rid of or smush together these constants and reduce the number of variables we have to play with. We're getting closer here. The next hurdle we need to overcome is this ES term that I've highlighted in pink. We don't know the concentration of ES. That's a very small number, and it's going to be hard to measure. So how do we fix that? Well, we can add this ESS term to both sides and then group these pesky ES's together. That will look like this. Plus ESS, and this will all cancel out. Plus ESS. Now, once we do that, we can again factor out that ES term once again. And this is what that will look like. ET times S equals Km plus S times ES. Since the ES term is the least defined, let's try to isolate it into one side of our equation. And we can achieve that by dividing both sides by this Km plus S term. Giving us ES equals ET times S divided by Km plus S. All right, it's getting simpler, right? However, our goal at the onset was to create an equation that will solve for reaction rates. ES is not the reaction rates. But if I told you that the initial rates of an enzyme's reaction, which we'll call V naught, is simply the product of the K2 term and ES, we can do another substitution. We can substitute ES for V naught divided by K2, just by dividing both sides by K2. So we can insert that into this equation and rearrange again. So we get V naught divided by K2 equals ET times S divided by Km plus S. And we want this Y term to be clean, so multiply both sides by K2, and there you go. And oh my god, we just got rid of all those K's, and now we've added K2 back into our equation. How the hell do we get rid of fats? Well, remember from last slide when we stated that V naught, the instantaneous velocity of our reaction, equals K2 times ES. I want you to consider this rate law in a very special scenario. Remember the enzyme kinetic curve I showed several slides back? It looks like a hyperbola, concentration of S on that axis, and reaction velocity is on the y, and it changes the velocity as we increase the concentration of substrate. Notice how the rates will approximate a constant value, the maximum possible velocity for the enzyme, called Vmax. Well, when the enzyme is saturated with substrate at these extreme values, V naught will equal Vmax. And at these extreme quantities of substrate, extra extreme quantities of substrate, what we will do is force every enzyme to be saturated, meaning there's no free enzyme. ET therefore will equal ES at this part of the curve. So we can take this generic rate law, V naught equals K2 times ES, and make it Vmax equals K2 times ET. And look what we have over here in hot pink. K2 times ET. This will allow us to do a substitution of this term, where K2 is going to be kind of tricky to find, I guess. And we can swap it in for Vmax, something that we can obtain graphically, yielding us V naught equals Vmax times the concentration of substrate, which we know because we prepared the solution of this fictitious experiment, Km, which is a constant unique to that enzyme, plus the concentration of substrate again. So here we have it. This is called the Michaelis-Menten equation, which can describe the rate of any enzyme as a function of its substrate at any concentration in theory. You do not have to memorize the entire derivation that we just did, but you do need to know this equation and the assumptions required to get us to this point. The Michaelis-Menten model is a simple but beautiful way to capture this complicated reaction equation that we started out with and reducing it into a relatively tame formula. And all that Maud Menten and Leonore Michaelis did to get there was a little bit of algebra. There was no trigonometry or calculus or differential equations, just basic add, subtract, multiply, divide, along with 5 simple but essential assumptions. Maud Menten, by the way, is one of the coolest scientists ever. There was an article written by Rebecca Skloot about her. Rebecca Skloot wrote The Immortal Life of Henrietta Lacks, by the way. And the title of the article is Some Call Her Miss Menten. It's a really short read. I think it's 4 pages. Highly recommend you Google it and read it on your own time because Maud Menten is a badass. And if this derivation wasn't proof enough, I'm sorry for you. Anyway, the Michaelis-Menten curve, shown over here, this will describe a lot about an enzyme's activity and its personality, if you will. I've mentioned this before, but take note on how this curve tapers off at extreme values of substrate. It asymptotically approaches this dotted line, which is Vmax, the theoretical maximum velocity of the enzyme. And you can visualize that here, and you can see that the tangent instantaneous velocity, as we move it along this curve, it's going to approach that value. So around here, we see V naught approximating Vmax. Another important takeaway is the Michaelis-Menten constant itself, Km. Recall that the units for Km are in molar, and that's important because it's going to describe the saturability of that enzyme for that particular substrate. What does that mean? It's the amount or concentration of substrate necessary to reach 50% of our enzyme's maximum velocity. If you're more of a math person, try this. Plug in S for the value of Km and see what it works out into. These two S's combine together to form 2S. The S at the denominator cancels out with the S in the numerator, leaving us with Vmax divided by 2. So the instantaneous velocity of a solution that has a substrate concentration equal to Km will give us a speed that is 1/2 of the maximum. Rewind that and try to work that through yourself. It's a little bit of a tricky concept at first blush, but it's really crucial. Km is the amount of S needed to reach 1/2 Vmax. What does this remind you of? Some people like to call the Km the binding constant of the enzyme for that substrate. And I see the similarities there. We're dealing with a hyperbolic curve, and the concentration of substrate needed to get to the halfway point of the maximum value is going to be halfway up this hyperbola. I get it. However, it's not a fair comparison. Conflating Km with Kd is a very big misconception. Because enzymes don't just bind substrate, they process them and spit them out as products. Nonetheless, the lower an enzyme's Km is, the higher is the enzyme's saturability. Less substrate will be needed to reach that top threshold. In the prehistoric era, scientists did not have the luxury of using software to accurately compute Km and Vmax based off of the curve. Computers were not around for general use until 70 years after enzymology was discovered. But a quick and dirty go-around to solving these crucial parameters was called the double reciprocal plot. Sometimes it's called the Lineweaver-Burke plot. And basically how you get it is you take your Michaelis-Menten expression and you divide the y term and the x terms into one. You invert them and you'll get something like this. And this expression is a line. We turn the hyperbola into a line. And lines are much easier to do manual calculations off from. So you can backcast your line with the ruler or math and solve for the x-intercept, and that will correlate with the inverse of the negative Km. And the slope of the line is equal to the Km divided by the Vmax. So having the x-intercept and the slope will let you calculate your Michaelis-Menten parameters. The problem here is that this method is not very accurate. Excel can do a really good job even of fitting a hyperbolic curve and getting these numbers way more accurately. There are many issues with the Lineweaver-Burke plot. We'll talk about it a little bit more in lab, but basically it's just really hard to get high values of substrates to make a nice linear Lineweaver-Burke line. And the extreme points of the Lineweaver-Burke plots are going to be very susceptible to error in particular because you're taking the reciprocal of your measurements along with the errors of those measurements, but we'll get more into that later. I just want you to be aware of the double reciprocal plots. I think it's mentioned on the MCATs, but in lab and in practice, you're probably never going to use it. We covered a lot of ground today. The Michaelis-Menten equation, I really think, is absolutely breathtaking. I mean, we have a complicated system of an enzyme whose rate law can be first order or second order or a mixture of the two, yet we found a way to calculate the rate of that enzyme just based off of substrate alone. That's huge. And that curve that we get, that hyperbolic plot, we plot substrate versus velocity, this will allow us to derive very important parameters that will measure the personality traits of said enzyme. Km and Vmax can be used to help us compare one enzyme to another. Is this enzyme more efficient? Is that enzyme more efficient? That's a useful skill, and we will unpack that further next time. Additionally, those Michaelis-Menten parameters will let us compare enzyme performance before and after activation and inhibition of that enzyme. All right, let's say you're trying to drug an enzyme. You can quantitatively measure how much your drug shuts it down based off of how badly that drug messes up your Km and Vmax values. And there's more applications of this knowledge beyond that, so I will end this with the following request. Stare at this equation and wonder.